So, thank you for the invitation, for the organizers. A lot of fun to be right after lunch. So hopefully some more food for thought. What I will try to go through is some of the examples, hopefully pressing on or opening some questions that we can discuss in a discussion in the afternoon that relates to aspects that algorithms are obviously interesting, important, and essential, but a lot of really new breakthrough analytical results come from breakthrough new data that is already long time existing algorithms have been applied to. So one of the things definitely for systems biology or integrated computational biology what I more prefer, because what we try to do is to integrate different kinds of data, different kinds of omics data, imaging data, clinical data, annotation data, medical records, information about the drugs, their target, transcriptionally modified profiles, and so on. And it obviously is represented in multiple different ways across the globe. Integration sounds simple, but it's not as simple because even simple thing as unique identifiers, obviously biologists and clinicians didn't go to database course 101 and they don't like unique identifiers in general. And there are all sorts of other issues that one has to deal with before we can move to the next level, which is trying to integrate any of this data through machine learning, data mining, statistical tools and identify what is potentially related to some phenotypes. But it's really the crux of understanding the biological mechanism of it is to linking it to different kind of networks. And networks can be obviously metabolic protein interactions, regulatory networks, drug target networks, and so on and so forth. So you can view this as multiple layers of networks and obviously multiple layers of this data that we are trying to integrate and analyze. Obviously, again, multiple machine learning data mining approaches to identify patterns in this. And the more important aspect is to interpret these patterns. Is it just something occurring or it has a clinical or biological meaning? Can we predict something that we didn't know, or are we only confirming what is already there? So through generating hypotheses and then validating, many times through clinical models, sometimes building really differential equations for modeling certain signaling cascades, we can identify what percentage of predictions were true or false. There are multiple aspects that one has to consider, and as in statistics, obviously once you have data, you can find statistical tests that will prove it significant. Somebody tells you that, oops, the labels were the opposite direction, and statistician could easily reply that, and that's even easier to prove that it's significant. So one has to be careful that what we see is really true. Obviously, it was already discussed many times today in terms of the randomizations and proper controls and so on. But one has to also consider other aspects in terms of the biases that we may not even know about. We talk about certain cancers, we talk about certain histological subtypes, certain grade stages, mutations, and so on. But there are so many things we will never know, and we cannot consider these groups to be homogeneous by any fact. There are many unknowns also. One of the interesting things, which obviously I'm not going to talk about, but it's chronobiology. And chronobiology basically looks into what parts of the proteome and genome are active in what state of cell cycle and also circadian rhythm. And so when you consider different responses for patients to chemotherapy, one of the reasons could also be that some patients are treated in the morning, others are in the afternoon, others are, and so on and so forth. So there are many variables that contribute to the variability of the results that we are not even considering. There's also a lot of missing data, various approaches to impute the data. But in most of the cases, you can find situations where you take the missing data as a special value, and you can find that the value is actually predictive. So then there might be a reason for the missing value, and you cannot ignore it. Many bad platforms and so on, we don't need to go there. But one of the other aspects I hopefully will have time to touch on is mistakes and so on, and kind of what is really ground truth. Today there were discussions about ES2 hybrid, mass spectrometry, curated interactions, and so on. And people have their favorites, but you can find errors in all of those. So you cannot blindly just say because it's a ES2 hybrid, it's a noisy data. Because sometimes mass spec will be a lot more noisy than ES2 hybrid, and vice versa. So one of the things with the integration is, and the beauty of this kind of analysis is that you can actually make the whole process completely blinded. You don't need to know what is the protein in the center of interest. You don't need to know what are the interacting partners. And you can start actually doing analysis in terms of prioritizing where the next experiment should go. You can have different thickness of the edge depending on how much confidence you might know is on the given edge, high confidence versus low confidence, or multiple pieces of evidence for interactions for those proteins. You can expand this first level of neighbors, and then you can start exploring the neighborhood of those interacting partners. So you can find clusters that share the same neighborhood, or you can find individual proteins that are very different from the remaining parts. Obviously, you can start adding additional annotation, for example, disease. If that gene, corresponding genes, is overexpressed, or protein within specific disease, that would be the height of the node. If it's another disease, it can be the width of the node. So obviously, you can easily identify proteins that are relevant to both diseases, or one or the other disease. You can obviously annotate simply with a gene ontology, and you already have a lot more information to prioritize where potentially next set of experiments would go, especially if you know what the phenotype is related to the protein of interest that you have in the middle. And particularly doing this kind of analysis with biologists, I find it very useful not to tell them what the protein is, because immediately they recognize certain names and they're excited about it. If it's FHL 12604, nobody wants to spend any money and time doing those experiments, yet that might be the best target to go after. The next level, you can obviously add the drugs. So we can identify drugs that target many of the interacting partners of the protein of interest, or drugs that are targeting, many drugs that are targeting the same protein. And again, the relationship is that from other literature, you may know which drugs are relevant to a specific disease. So you have already disease annotation here. Now you have another piece of evidence from completely orthogonal type of data to tell you that that potentially is a relevant piece, and maybe this would be the set of interactions and the set of experiments that you can start planning in that direction. So one of the kind of follow-up examples is, for example, recently we were doing sequencing analysis of lung cancer samples from Brazilian population and identified differential microRNAs. Some of them were novel, some of them were already reported in the literature in lung cancer, so we can start debating, is it, because this is the first cohort from Brazilian population, if there is some specific microRNAs related to this population. But then you can link them to transcriptional factors that potentially are regulated by those microRNAs, or other prognostic genes in lung cancer that are potentially regulated by these microRNAs. Through simple network analysis, you can identify those that are targeted by many of those differential microRNAs that are overexpressed, and thus very easily, through integrating these multiple pieces of information from different resources, you can highlight which genes potentially are worthwhile validating in this specific experiment. What is really neat is that you can take those genes then and take yet another orthogonal data, protein interactions, and basically link them through physical protein interactions. Now, the reason why it's interesting that we started with microRNAs, we predicted microRNA to gene targets, annotated with transcriptional network, gene ontology, and prognostic and differential genes in lung cancer, and then took what came out of it, put it into the network of protein interactions, and then you can actually do simple statistical enrichment of, are we finding enrichment in this network now for the proteins that are known to be prognostic? So there is noise in all of the data. There are missing pieces in all of the data. But once you start finding significance with the proper, obviously, controls and false discovery rate controls, that shows that there must be some biologically and clinically relevant signal that you can follow up on. There were also discussions about which interactions are good or bad, or which high throughput assays is good or bad. I like this network figure also for the fact not only that it's nice and colorful, green, blossoming, but also when you look into all of the different colors of the edges are different methods, biological assays that were used for detecting these interactions for these two melatonin receptors. And you don't need to kind of analyze it in any specific way, but you see that there is a lot of heterogeneity. If you would use just one of the methods, you have small fraction of the interactome that is there. So it's really multitude of methods that you have to integrate in order to have a better sense of what these receptors or how they are interacting. And one of the reasons that was pulled out in a paper is, for example, to identifying the mechanism. The thick edges are from the paper interactions that were detected through these methods. So it's only through this new interaction that you can explain some of the mechanism that was already postulated in the literature, but there was no direct link between these receptors and the voltage-gated calcium channel. So again, by integrating different kinds of assays is the only way how you could explain some of the known biology. So one of the aspects is that when we are doing this kind of integration, we usually have some either biological or clinical reason to do those integrations, and they will drive our interest in integrating the data and possibly validation. But we need to study relevant biology. We cannot study just what is significant. We always need to kind of keep track of, does it make sense? Can we do in vitro functional assays that will prove it or not? And one of the aspects is that if you take, for example, prognostic genes, you can map them to the network and start studying what processes they are involved in, what are the connections between them you can identify on the networks. And to really highlight the aspect of data does matter more than the approach is if you take just one of the data sets, not integrated data, you have significantly more sparse network. You may apply any kind of sophisticated graph theory algorithm, you get completely different results from these two networks. And in one, you cannot really start planning any follow-up experiments. In the other one, you may start planning experiments. There is possibility that some of these edges are wrong, but you have the reason and also the context of how to validate them, where to validate them, in what cells, and what's the... There are different assays that you can also apply once you have an edge, as opposed to when you are searching for possible edges. Obviously, eventually we will start to, or we are already starting to have another problem that almost everything will be connected to almost anything, or the density of this network is going to be larger and larger, and we have to be careful. There were already examples about UBC. Human network now is about 800,000 edges and 18,000 proteins. I will have some example on it, but also what you can do with it. So one aspect is that obviously UBC and P53 was already discussed. Should we remove it because it's connected to everything, or should we consider it, but then it will be showing up in almost any analysis? So scalability of the algorithm is definitely one aspect. If you want to visualize it, analyze it, run these algorithms. But also then quality of the data and annotation of these nodes and edges that will enable you to filter out in and out what is relevant and what is not. So I will go through this faster. Again, it's kind of continuing motivation is that we really have to integrate multiple hybrid methods, not only different biological assays, but also computational methods that will help us enhance what we can learn from this network, how can we analyze them to the disease-related or biology-related context. So several papers now. This is the new interaction database, as I was mentioning, almost 18,000 proteins and a little bit over 800,000 edges. But the big chunk, which is the blue, are the predicted interactions. It's multiple methods using, in February I was talking in more detail about it, but the papers go into detail of what information you can include from sequence, from expression, from homology, and so on. Orthologs or homology-based predictions are a relatively small fraction of these. And still, even when you integrate what is known from high-throughput experiments and from curated literature, it's more than other resources. Now, you can still argue that this chunk is going to give you a lot of false positives. There is a lot of validation that not necessarily that's true. We're validating with other biological assays. True positives versus false positive is very comparable to the biological assays. So it's not much worse, it's comparable. The other aspect I like to kind of highlight, and this is relatively limited set that we have been doing about 200 pairs of interactions. People believe that what is curated is correct. By chance, we found a couple of incorrect interactions, and then we started to systematically go through it. And this just shows a highlight of some of the errors. The blue ones are errors. The green ones are correctly curated data. Obviously, different source of the database have different ratio of correct and incorrect. Some have more incorrect than correct. One has to be careful about that. But in general, it's showing about 50% of errors, even in curated data sets. So you cannot say that if it's a curated resource, that's the high confidence interaction. If it's yeast-2 hybrid, it's low confidence. Those generalizations just don't work.
What are the true positives in the example above?
Here?
No.
Oh, here?
Yes.
So this was, this is the, from Nature Methods a couple of years back. And what they had was 92, the caveat of that analysis, that was only 92 positive pairs, 92 true negatives. And they were running 5 different assays, also not running mass spec and not running other methods. But still, basically, what is the sensitivity and what's the false discovery rate for those individual methods? And then we ran the prediction algorithm that was able to control the false discovery rate and still detected twice as many interactions and true positives as the other assays. But it's only 92. And also this, obviously, is only 200. So would be interesting to plow through it in more systematic way. So going back to the validation, really the power is the integration. I mean, I showed one example where you integrate mass spec, yeast-2 hybrid, mammoth screen, different biological assays, and that gives you much better sensitivity for the screen. In this case, we were linking computational predictions with biological assay, and again, it gives you much more sensitivity and controllable false discovery rate. Obviously, the biological assay also matters, which is kind of interesting that going back to the comment at the beginning is what is the ground truth, right? When we make a prediction, the biological experiment is the ground truth. But if I give my predicted interactions to Vidal, who is running his screen at about 25% sensitivity, well, gives me from 100 interactions, give me 25. That doesn't mean that the rest is incorrect. It's just that's the sensitivity of that screen. So it's very useful to also be able to potentially identify, should I give this interaction to Paulson? Should I give these interactions to Uli? Or what is the method, what is the lab, what are the cells in which I have the highest chance of validating the interaction? So true positive, false negative. So going back to this interaction database, obviously the numbers are interesting, but this is starting to be more and more useful. As I was mentioning, that the networks are denser and denser, where we try to annotate the interactions with different tissues based on a gene and protein evidence. Obviously, for some organisms, there is very limited evidence available that we can integrate at the present time, but for humans, it's relatively well characterized. So what this can give you is that you can go through the tissue and identify where those interacting pairs are detectable or conserved. And you can also then, a lot of the human cancer genes or biology is being done on a mouse model or on a fly organism. So you can also determine if it's the right model for whatever interactions or part of the pathway or complex you are studying. And you can, I don't have time to go into much detail, but you can actually identify those where these interactions are conserved. So not just that you have homology of the corresponding proteins, but that interaction is likely also existent in a given tissue. So how can you use such information is, it's still under review, so I don't want to go into too much detail on that, but suffice to say, proteins of interest, you generate a network that potentially connects them because there is a lot of interest. In this case, it's a lung-to-brain metastasis. So what makes sense is to not consider all of the interactions, but consider interactions that are lung, brain, or lung-on-brain specific. I'm not interested in what potential is in spleen and prostate and so on. So we can easily identify what subset is relevant to the analysis. And what is really cool, again, as call it a validation output, is that, sorry, let me backtrack here. So those that are highlighted here, we identified as being central in the network through simple all-pair shortest path analysis of centrality in the network. And then what is interesting is that actually 18 of the 20 identified through this method in lung adenocarcinoma have a good hazard ratio and a significant value. And on top of it, when you combine them in a signature in a regression model, on another that we built on a TCGA data, we were able to validate significance of that split of the patients with a strongly predictive or prognostic value. So it kind of links, again, information that it's a large network, it's noisy, but the level of tissue specificity that we can interject into it has a clinical implication because we identified proteins that actually validated quite nicely, 18 out of 20, clinically. So another kind of aspect is that you can not only integrate, obviously, gene ontology, you can integrate disease information, you can integrate biological processes where these proteins are interacting. So again, work with Igor's, Steiger's group, identifying individual novel interactions and then characterizing them in terms of where the links are more than you would expect by random chance, that those multiple interacting proteins are involved in the same disease, they are involved in the same process, they have similar biological function, and in a sense enrich for that. Which, on the next level, you can link information from what we know about drug targets and also what we know about transcriptionally regulated genes as a result of application of the drug. Because all of those genes that we know for a given drug are changed transcriptionally after application of the drug. Through the novel interaction, we can actually now link them. Because obviously, if drug targets this protein and these genes change, there must be some path that goes in between them. So again, this kind of highlights that those new interactions are the bridges or tunnels, or whatever you all call them, roads, that help you link what we know is changing and what we know is a drug target. So now, kind of interesting aspect. What we wanted to see is that is there more information, in a sense, in these networks? And it has been described here and also in similar kind of meetings that you take expression data and you can generate co-expression graph based on differential expression across two conditions. In this case, normal lung and lung cancer. As Natasha, many years, actually scarily over a decade ago, introduced the approach with graphlets analysis, what is intriguing is that then you take these large graphs, and in a sense why it's useful is that this is just lung cancer. You can then apply it to any different cancer, any different disease, and a lot of graph comparisons. But you can simply look into the frequency of individual graphlets and identify which graphlets are more frequent in a tumor graph versus the normal graph. And what is interesting is that it seems, which is not that surprising, that tumor graph is losing all of the graphs or many of the graphlets with cycles. So it's losing the redundancy. And also what is interesting is that long stretches of linear paths are basically potentially creating shortcuts for those individual graphs that are, in a sense, are connecting maybe pathways that are normally not connected. So we started to explore it further, and it's definitely true that you are linking pathways. And again, it was discussed earlier, what is a pathway? Doesn't exist in nature. We kind of identified as, yes?
The tumor graphs, are they inferred from the tumor data, or are they projections of normal graph onto the tumor transcriptome?
No, it's normal samples and tumor samples. And from those, you generate the co-expression graphs.
Together?
No, separately. And then you identify what is different and what is similar between them. But it's kind of the first step. Obviously, there are the caveat with this approach. It's almost the same as when we had, where's Ben, the hotnet, right? It's not that this is wrong, but there's a version 2 that is coming. Because one of the aspects is that this is looking at the co-expression. And that doesn't mean that those are corresponding to protein interactions. So it's interesting that you can find these differences, but the next step was to see how does it overlap with known or predicted protein interactions. That step makes sense kind of only after the protein interaction network is relatively large. Otherwise, you get too many false negatives. So the next step is to see if what we identify as potentially disrupted, can we apply this, call it a mathematical approach, and see if we can repair the network. Most of the drug prediction algorithms basically work to identify individual nodes and try to change the expression of the nodes. That's fine. But now we actually want to change the whole network. So the optimization process then has to go into identifying what do we know about the node in terms of the data that we analyze, and also data, in this case, across large number of lung cancer data sets, how it's upregulated or downregulated significantly. And also, what do we know about the edge? Meaning that if we have two nodes linked, are they correlated positively or negatively or not correlated at all when we are considering the normal and cancer set? And we also add the additional information that do we have any evidence that those corresponding proteins are interacting pair, or at least predicted to interact. And so we are obviously trying to identify what is biologically meaningful, and thus we want potentially true physical protein interaction, not just co-expression. But just co-expression is also useful because we know we don't know all of the protein interactions. So then the optimization on a drug level is that not only we want to find what is the most interesting subgraph based on this weighting, but also we want to find the minimal number of drugs that will maximally change the graph as we want, both on the nodes and edges. What is then basically the step is then you can really find the optimal settings of what drugs, what combination of drugs, where do they apply, and what should they change. And you can really quantitate it based on an impact weight from which you can very systematically pick up combination of drugs that would be working for that subgraph. And luckily then in a biological lab we can try on, in this case, 3 different lung cancer cell lines and identify that not only they change the phenotype of the cell lines, but also the drug combination works better than single drugs individually. And in most of the cases that we tested in all of these 3 different cell lines. Now, what is also interesting is that you can go mechanistically back to this graph and identify, did we change the node, and did we change the pairs of the nodes? So in a sense, did we repair the network as a result? And in most of the cases, we actually did change majority of the subnetwork of interest. So obviously, limited in vitro validation, but at least it kind of points into the aspect that we might be able to start rewiring the networks and make them more similar. I mean, take the lung cancer network and make it more similar to normal lung network, and thus potentially have some drive into the cure.
Can you explain better what do you mean by rewiring? In what sense does the drug combination not rewire the network?
Because you are not only considering that, I mean, the traditional approach of drug repositioning is that you change the expression of the node. Whereas here we are looking also in all of the connected pairs of the nodes, how are we changing the other node? Because drug might be target or transcriptionally changing this node, but it's linked to this node, and it's changing as well. So you have to consider, are they changing in the same direction, opposite direction? You may fix one, but you may screw up the other. So you have to consider all of those nodes together as a group, as a signature, in a sense.
But if it's just one drug per one node, then the most you are changing is 1 or 2 edges.
Yeah.
But what's the model that's underlying there? I don't understand. How do you predict how the drug affects?
So that comes in the earlier, actually that's a good point, I don't hear. So through other data we know what are the drug targets. So we can map drug targets to the nodes within this network, or we can map drug that we know transcriptionally modify corresponding genes. And so based on those 2 pieces of information, you overlay it on the network and identify what do you need to change. So I will try to go faster. I don't know how many people follow Dr. House. One of the things that is kind of intriguing is the experimentation he does. He doesn't, in a sense, comply with the regulations and so on. But from the diagnostic viewpoint, makes more sense because you have to perturb the system in order to understand what potentially is wrong. But the other aspect that kind of comes from that series is that everybody lies. I mean, all of the patients lie. And kind of the motivation for it is that, well, in a lot of the clinical work or clinically related work, again, ground truth, we take the literature, published literature as ground truth. And unfortunately, a lot of it is actually false. And you may say, well, what is lot? So a few years back, we were doing analysis in terms of how many papers are retracted. And there is exponentially, I mean, there is an exponential increase of retractions, even when you consider how many more papers are published every year. If you take the titles of those papers, strikingly, there's a lot of clinical and biology and patient-related studies that are being retracted. So we repeated the analysis in December, and I mean, there is little drop, but it's still a highly exponential increase in number of retracted papers. Still, the same kind of aspect when you look into the titles or journals, there is a lot of clinically relevant journals that are being retracted, or papers that are being retracted. And when you consider it's a 5%, that's a relatively large number. So there were other studies, this is a few years back now, and what is interesting is kind of two aspects that kind of come from it. It's already bad that these papers are retracted, but there is a large fraction of fraudulent or suspected fraudulent papers. There is only less than 25% of mistakes. I mean, less than 25% is still large, but less than 25% mistake. What is worse, though, that if there are errors, people get it in about 26 months in average. If it's fraudulent, it takes almost twice as long. And that's striking. I mean, you have smart people who fabricate the data or change the experiments, and it takes us a long time, I mean, as a community, to find those errors. This is not our data. Somebody else did this kind of analysis. But that shows you that you cannot trust everything that you read. You cannot trust all of the curated interactions either, and so on. So to kind of finalize, I hope I convinced you that diversity is necessary. We have to have multiple methods, computational and biological assays, because only through those we will be able to explore wider range of clinical and biological phenomena. Putting together biological and computational assay obviously makes faster, cheaper, and more relevant predictions and validations of those experiments. There are many aspects that we don't know yet, and we have to be open-minded. We cannot have biases that this should be like this. Many times we would be wrong. And I don't have time at all to go into this, but there is some cool data that we now have where people are focusing, again, on drug repositioning, treatment of cancer, and so on, or other diseases. And there is a lot of stuff from what we eat and what we do that affects it probably even stronger. So with that, thanks to all of the different collaborators across different diseases and funding agencies, other lab members that I didn't mention before, and if people are interested, these tools are publicly available. Thank you.
A couple questions and then we should.
So the analysis of network in tumor and normal, that you thought about the issue of heterogeneity that might make it
Heterogeneity in terms
that will make it seem like dysregulated, but it's actually just
I mean, if we analyze it across multiple different, the training data was multiple data sets, and we validated it on multiple validation data sets. And when you see the same pattern across different cohorts, that diminishes the potential problem that there would be heterogeneity that would play a significant role.
patterns in terms of specific genes, yes, but in terms of general dysregulation, no. That doesn't tell us anything about it.
No, it does, because you would not see anything. I mean, there will be multiple heterogeneities that will be present, and very unlikely you will get the same co-expression differences between the normal and different heterogeneous subpopulations. But to answer the other kind of part of the question is that, or the aspect is that, yes, you can do this kind of analysis not only on cancer type basis, you can do different histological subtypes, and we did actually even analysis of, for example, in lung cancer context of KRAS-specific mutation adenocarcinoma. So you can definitely zoom in on much more purer subpopulations. But usually what that means is that you have much smaller number of samples. So again, one of the aspects of larger scale analysis, you have larger samples, you have more power, but some differences in terms of the cohorts or heterogeneity. You zoom in on a more pure population, but you have smaller number of samples, and you might not have enough for the independent validation.
So maybe we'll take additional questions at the break, and thank you again.